I want you to start this deep dive by doing something uh, very low-tech. Just lick your arm, pick a spot on your skin.
Okay.
It looks pretty static, right? Just a skin.
It's doing its job. It's, protecting you, keeping the outside out and the inside in.
Right. It's a boring adult cell. It has signed a contract to be a skin cell, and it's going to stick to that contract until it, dies and flakes off. But imagine for a second that we could take a microscopic sample of that skin, put it in dish, and just sprinkle some, chemical dust on it.
And we aren't talking about moisturizing it or exfoliating?
No, not at all. We're talking about time travel. Imagine watching that skin cell physically morph. It stops being skin. It travels backward through its own biological history.
Wow.
It transforms into a glowing, branching tree capable of becoming, say, a beating heart cell or a firing neuron or part of the liver.
Okay, that sounds like the opening scene of a sci-fi movie.
Oh.
Or, alchemy.
Exactly. But the core question we are tackling today is this alchemy? Can we actually rewind a cell's biological clock? Or is this just hype?
It is very much reality. In fact, it's not just a theory. It is a Nobel Prize-winning scientific fact as of 2012.
Right.
So we aren't just talking about, dreaming of youth. We are talking about a proven technology called cellular reprogramming.
So today, we are going to unpack the discoveries of two absolute giants in this field, John Gurdon and Shinya Yamanaka.
The grandfather and the hacker.
I like that.
Oof.
We're going to figure out how on earth we can turn back time at the cellular level. We're going to look at the massive medical benefits, which go way beyond just anti-aging, into things like disease modeling and organoids. But we also have to look at the cold shower reality check.
The catch.
There's always a catch. The specific, dangerous risks like cancer that keep eternal youth just out of reach. For now.
It's a story about breaking the laws of biology, or at least the laws we thought were written in stone.
Let's start with those laws.
Yeah.
Because to understand why this is such a big deal, you have to understand the rulebook biology was playing by before, 1962?
Yeah, before 62. The dogma was called the law of irreversibility. It's actually pretty intuitive. Think about growing up. You start as a fertilized egg, a blank slate.
Total freedom. You can be a brain, a toe, a spleen, whatever.
Anything. But as the embryo develops, cells make choices. You become a nerve cell, a gut cell, a muscle cell. And the prevailing wisdom was that once a cell chose a path, it was locked in. Forever.
So a nerve cell stays a nerve cell, you grow up, you don't grow down.
Precisely. The cell differentiates, and it basically shuts down all the other options. It locks the library doors to all the other genetic instructions.
Until John Gurdon came along in 1962 and decided to mess with some frogs.
John Gurdon is the first protagonist here. In 1962 at Oxford, he designed an experiment that is just beautiful in its simplicity, but it was considered absolutely heretical at the time.
So what did he do?
He took a specialized somatic cell from a gut cell from the intestine of a fully adult frog, and he extracted the nucleus.
Okay, let's pause on that. The nucleus is the command center.
Yeah.
Right? It holds all the DNA.
Right. The instruction manual. So he has the manual from an adult intestine, the manual that says, I digest food. Then he took an unfertilized frog egg and he used UV light to destroy its nucleus.
So he has an empty shell, a blank egg with no instructions.
Correct. He then injected the adult gut nucleus into that empty egg. Now, if the old rule book was right...
Nothing should have happened.
Right.
Because the DNA came from a gut cell. He should have just sat there confused or maybe tried to build a tiny piece of intestine or something.
That's what everyone expected. The DNA should have said, I'm a gut cell, I don't know how to build a whole frog. But that's not what happened.
What happened?
The egg developed into a normal swimming tadpole.
That must have been a huge shock to the scientific community.
It was earth-shattering. It proved that the adult DNA hadn't lost the instructions to make a whole frog. It hadn't deleted the files. Exactly. And the environment of the egg, the cytoplasm, it had the keys to unlock them. It just reset the gut cell back to zero.
Okay, so that was 1962. We knew it worked in amphibians, but, humans aren't frogs. And I feel like there was a long gap before people really believed this applied to us.
A massive gap. We're talking decades. Skeptics were saying, sure, frogs are simple. They can regrow limbs. Mammals are complex. Mammals are different.
And it took until 1997 to bridge that gap, which brings in the most famous sheep in history.
Dolly.
I remember when the news about Dolly hit, it felt like the world just stopped. It was everywhere.
It did. Ian Wilmot and Keith Campbell basically did Gurdon's frog experiment, but with a mammal. They took a cell from an adult use utter...
A mammary gland cell.
Right, and they transferred its nucleus into an empty sheep egg.
And out came Dolly, a clone.
A genetic copy of the adult you. It proved that cellular reprogramming wasn't just a frog trick. The law of irreversibility was broken for mammals, too.
So here's the question I think a lot of people had in the late 90s. If we could clone shape, why didn't we see, like, rejuvenation clinics opening up in 1998? Why didn't this immediately revolutionize medicine for humans?
Two big reasons: efficiency and ethics. The method was incredibly inefficient. I mean, to get the one dolly, they had hundreds and hundreds of failures.
So it's not like a factory line where you press a button and get a result?
No, not at all. Cloning is difficult, it's expensive, and it has a staggeringly high failure rate. Most of the embryos just died or had severe abnormalities.
And the ethics part.
Well, to do this kind of therapy on a human, say to grow you a new liver, we would need to create a cloned embryo of you and then harvest the stem cells from it.
Which means creating and then destroying a potential life.
Right. You're getting into the destruction of embryos, plus the logistical nightmare of just sourcing human eggs. It was a minefield.
So the science was there, but the application was impossible.
Pretty much. We needed a workaround. A way to get the result, the young, flexible cells, without the egg and without the embryo.
Which brings us to the second Nobel laureate in our story.
Shinya Yamanaka. If Gurdon was the explorer who found the new continent, Yamanaka was the hacker trying to crack the code.
This is 2006. What was Yamanaka's approach?
He looked at the egg and asked a software question. He basically said, I don't need the whole egg, I just need to know which genes the egg is using to reset the DNA. He wanted the source code.
He wanted the password to the computer.
Exactly. He hypothesized that there must be specific transcription factors. These are proteins that turn genes on and off that act as the master reset switch.
How did he even find them? Did you know what he was looking for?
He started with a bit of a brute force attack. He identified 24 candidate genes that seemed to be active in embryos, and he put all 24 into a skin cell, and it worked. The skin cell turned into a stem cell.
But 24 is a lot of variables for a clean medical therapy.
Way too many. So he started removing them one by one. You know, if I take this one out, does it still work? If I take that one out, does it fail?
The process of elimination.
He just whittled it down. And he eventually found that you didn't need 24. You only needed 4.
Just 4?
Just 4 genes. Octa4, SOX2, KLF4, and C-Mike.
That sounds like a boy band. The Yamanaka factors.
In the biological world, they are absolute rock stars. By introducing just these 4 factors into a mouse skin cell and later human cells, he was able to reprogram them all the way back to an embryonic-like state.
Let's just clarify this because it's so important. No egg involved.
No egg, no embryo destruction, just a skin cell, a petri dish, and these 4 factors.
That is the aha moment. That's the absolute game changer.
He called them induced pluripotent stem cells, or iPSCs. Induced because we forced them to do it. Pluripotent because they have the power, potent, to become many pluri-things.
This changed everything, didn't it? Suddenly, you don't need a donor. You could theoretically take my skin cells, treat them with these 4 factors, and create stem cells that are a perfect genetic match to me.
Exactly. It's reproducible, it's scalable, and it's personalized. It just bypassed the ethical wars and the supply chain issues.
I want to pause on the mechanism here for a second because reprogramming is a buzzword. What is actually happening inside the cell? I saw a metaphor in our notes about a computer reboot. Is that accurate?
It's the best way to understand it. Think of a specialized cell, like that skin cell on your arm, as a computer that's been running for years. It has a specific operating system open, specific apps running.
Like make keratin or something.
Right, and a lot of background processes are locked down.
Yeah.
It's in adult mode.
Okay, it's cluttered. It's set in its ways.
But when you add the Yamanaka factors, you aren't smashing the hard drive. The DNA hardware isn't changing. You are triggering a system reboot. You are forcing the operating system to force quit all those specialized apps and restart in safe mode or, factory settings.
So the hardware is the same, the genetic code is still mine, but the software resets to the initial startup screen.
Precisely. These transcription factors, they physically restructure the chromatin, which is the packaging of the DNA. They open up the closed books and they close the open ones. They just shift the regulation from adult mode back to startup mode.
But just to be clear, this isn't like rejuvenating the soul of the cell. We aren't getting mystical here.
No, absolutely not. It's a mechanical change in gene regulation. Pure biological engineering.
Okay, so we have this technology. We can reboot cells. We can turn my skin into a baby state cell. This is where I want to really dig into the so what question.
Hmm.
Beyond the cool factor, what does this actually do for medicine right now? Because I don't see people walking around with Benjamin Button disease.
And that's a good thing, which we will definitely get to. But the immediate revolution isn't about making people younger. It's about understanding disease. The primary benefit we're seeing right now is something called disease modeling.
How does that work in practice?
Okay, imagine a patient with a rare genetic heart condition. Maybe a rhythm disorder that causes sudden cardiac arrest. Previously, if we wanted to study their heart cells to see what was wrong, we'd have to do a heart biopsy.
Which is invasive and dangerous.
Yeah.
You don't want to be poking holes in a sick heart.
Very. Or we'd study a mouse heart, which, it isn't the same. But with iPSC technology, we take a simple skin scrape from that patient. Easy, painless.
Mm-hmm.
We reprogram those skin cells into stem cells using the Yamanaka factors.
Mm.
Then, in the lab, we coax those stem cells to turn into heart cells.
So you have the patient's actual heart cells with a specific genetic defect beating in a petri dish.
Exactly. We call it disease in a dish. You can watch how the disease develops in real time. You can see precisely what goes wrong at the molecular level.
That sounds incredible for testing drugs.
It is transformative. Before you give an experimental drug to a sick patient, you can test it on their cells in the dish. Does it fix the rhythm? Does it kill the cell? You can test toxicity and efficacy without risking the patient's life.
And I've heard about something even crazier than cells in a dish. Organoids. It sounds like something from a horror movie.
It does sound a bit creepy, but it's fascinating. This is the next step up. We aren't just growing flat layers of cells anymore. Scientists are using iPSCs to grow 3D structures that mimic organs. Mini brains, mini livers, mini kidneys.
Wait, a mini brain. Are we growing consciousness in a jar here?
No, don't worry. It's not conscious. It's about the size of a pea. But it's a three-dimensional ball of neurons that organizes itself similarly to a developing brain. It gets layers. It has structure.
What do you do with a pea-sized brain?
You study things that mice just can't show us. For example, when the Zika virus outbreak happened, scientists were struggling to prove that Zika caused microcephaly small brains in babies.
I remember that.
They used brain organoids from iPSCs to see exactly how the virus was attacking neural stem cells. You couldn't see that in a 2D culture.
So instead of testing on animals, which might not react the same way humans do, we're testing on human tissue models.
Correct. It allows for better checking. It removes a huge amount of guesswork from medicine. It's a simulation of you, made from you.
That is fascinating.
Yeah.
It's like having a crash test dummy made of your own cells.
That's a very apt analogy.
Okay, now we have to address the elephant in the room. The listener's thinking it, I'm thinking it. If we can reboot cells to be young again, if we can turn an old skin cell into a fresh baby-like stem cell, why can't we just put these factors into a pill, take it, and reboot our whole body? Why isn't this the cure for aging?
This is where we have to deliver the cold shower. The process has two major barriers that keep us from the fountain of youth: control and safety, specifically the risk of cancer.
What goes wrong? Is it the factors themselves?
It is. Remember those 4 Yamanaka factors? OCT4, SOX2, KLF4, and C-Mike.
The rock stars.
Well, one of them is a bit of a bad boy. C-Mike.
It's C-Mike.
Yeah, what does it do?
C-Mike is a very, very powerful gene for driving cell growth and metabolism. It pushes the cell to divide and grow. But in the world of oncology cancer research, it's known as an oncogene. Oh, that's not a word you want to hear. That's a cancer-causing gene.
Exactly. C-Mike is heavily linked to many types of cancers. You see, pluripotency, that ability to grow into anything, is dangerously close to uncontrolled growth.
And uncontrolled growth is the definition of cancer.
Precisely. If you rewind a cell too far or too loosely, or if you leave that reboot switch on for too long, you risk creating tumors. Specifically, something called a teratoma.
A teratoma. I think I've seen pictures of these.
Yeah.
They are nightmares.
They are. Because the cells are pluripotent, they try to become everything at once. So you get a tumor that contains hair, teeth, bone, muscle, gut tissue, all just growing chaotically in a lump.
Yikes. So a cell that can become anything might decide to become a monster.
That's a good way to put it. The reboot is powerful, but it's risky. If you tried to systemically reboot a human being by injecting these factors into your bloodstream, you wouldn't get a younger human. You would likely get a human filled with teratomas and aggressive cancers.
So the line between young cell and cancer cell is terrifyingly thin.
It is razor thin. The body has very strict controls on cell growth for a very good reason. When we hack that system, we are playing with fire.
So this idea of eternal youth, Just drinking a potion and staying 25 forever, that's off the table.
For now, yes. We need to completely reframe the goal. We shouldn't be thinking about eternal youth. We should be thinking about eternal repair.
Distinguish those for me. What's the difference between youth and repair?
Eternal youth is that romanticized fantasy of whole-body rejuvenation. As we just discussed, that carries the risk of total systemic failure or cancer. Eternal repair is the realistic scientific practice. It involves cautious local repair.
Give me an example of eternal repair that might actually happen soon.
Let's say you have macular degeneration. You're losing your eyesight because specific cells in your retina are dying.
Right.
Eternal repair means we take your skin cells, turn them into iPSCs in the lab, carefully differentiate them into healthy retinal cells, and we make damn sure there are no cancerous ones left, and then we transplant just those cells into your eye.
So we aren't trying to make the whole organism a teenager again. We are just patching a flat tire.
Exactly. We're restoring specific functions. A heart patch for a heart attack victim. New dopamine-producing neurons for a Parkinson's patient. Real-world application is all about strict boundaries. We don't want embryonic cells running wild in the body. We want controlled restoration of function.
It seems like the difference between flooding a house to clean the floors versus just spot cleaning a stain.
Yes.
Flooding it might clean the floor, but it ruins the drywall and the electrical.
That is a perfect metaphor. We want to spot clean diseases, not flood the system with chaos. The goal isn't to live forever as a 20-year-old. It's to keep the machinery running smoothly for as long as possible by replacing broken parts.
So bringing this all back to where we started. In 2012, Gurdon and Yamanaka shared the Nobel Prize.
A very well-deserved one.
But it wasn't for a fountain of youth potion. It wasn't for a product you can buy at the pharmacy.
No. It was for the proof of principle. They proved that cell specialization is reversible, that the law of irreversibility was wrong.
That the clock isn't broken, just locked.
Exactly. We now possess the tool to move the cellular clock. We know the program isn't erased, and that is just a profound shift in how we understand life itself.
But the challenge now is the instruction manual for that tool.
The challenge is control. We have the ability to press the reboot button, but doing so safely in a human being is infinitely harder than doing it in a petri dish. We're essentially learning how to code with the source code of life, but we are still debugging.
It really leaves you with a provocative thought, doesn't it? We have this technology that essentially hacks the source code of human biology. We can rewind the tape, but the listener has to wonder, just because we can rewind, does that mean we should? And who decides when it's safe to press that button?
And how do we ensure the reboot doesn't crash the entire system? That is the question that will define the next 50 years of medicine.
It's a lot to think about. From frogs to sheep to a hacker in a lab, and now to the future of medicine. Thanks for unpacking this deep dive with us.
It was a pleasure.
And to you listening, keep asking the hard questions. We'll catch you on the next deep dive.